Hello and welcome to this eukaryotic cell biology video. I'm Dr. Aled Roberts, and in this video we're going to explore the history of the eukaryotic nucleus. Now, when we look at the eukaryotic cell, there are a number of differences which set it apart from its prokaryotic counterpart. However, one of the most influential differences is that the eukaryotic cell contains a nucleus. And so the nucleus is one of the most important membrane-bound organelles within the eukaryotic cell, and as well as its presence being a defining feature, it is of crucial importance to the cell because it houses the vast majority of the cell's genetic information, making it what we might call the brain or control centre of the eukaryotic cell, controlling all aspects of cellular life in one way or another. Now, in general terms, it occupies about 10% of the cell's volume, which is quite sizable and can be seen with the right dyes and a light microscope. Now, as I've hinted at, the nucleus is crucially important to the proper functioning of the eukaryotic cell. However, whilst the nucleus is important, the eukaryotic cell requires all other membrane-bound organelles and cellular components to function effectively. If we think on a much larger scale, the brain is the control centre of our bodies, but it can't see without the eyes, it can't smell without the nose, taste without the tongue, or walk without our legs. And the eukaryotic cell is no different. Whilst the nucleus contains the vast majority of the genetic information, functioning as a sort of brain, giving out various instructions, it needs the other organelles to read, interpret and make sense of these instructions so that the related biological process can be completed. Now let's have a look at the long history of the nucleus from when it was first discovered to our current understanding. And it all starts with the work of Antoine van Leeuwenhoek, often called the father of microbiology. Now, Leeuwenhoek wasn't a scientist by training, but he had this fascination with lenses and spent years crafting an upgraded version of the microscope, with increased resolving power, slightly more magnification through the use of powerful lenses, and a dedicated sample holder, allowing for better focusing of samples. Now, with this setup, Leeuwenhoek became the very first person to observe and document single-celled microorganisms through his famous Letters on the Protozoa in 1677. In this publication, he described what he called animalcules, which translates to small animals. Now, what he was actually saying was both protozoa and bacteria, but he didn't know what they were yet. Now, his experiments and observations literally changed how we viewed the microscopic world. The improved design offered significantly greater resolving power over the original compound microscope developed by Robert Hooke. In fact, it was so advanced that if it could visualize individual prokaryotic cells, it should in theory have been capable of resolving larger intracellular structures such as the eukaryotic nucleus, which exists on a comparable scale. This highlights just how remarkable his craftsmanship and optical precision were for the 17th century. Now, after the work of Leeuwenhoek, there was a bit of a wait before one of the next key discoveries was made, and this came a little over a century later, when we come to Franz Baur, a highly skilled botanical illustrator working in the early 19th century. Now, Baur wasn't formally trained as a scientist either, but like Leeuwenhoek, he had an eye for detail and a deep curiosity about the microscopic world, particularly the inner structure of plant cells. And so, using one of the most advanced light microscopes available at the time, visualized plant cells, describing and detailing a distinct, rounded structure inside the cell, what we now recognize as the cell nucleus. Now, although Baur didn't fully understand its function, his careful observations provided some of the earliest visual evidence of this organelle. Now, working independently around 1825, we come to Jan Purkyne, a Czech anatomist and physiologist whose work built upon earlier observations of cellular structures. And so, while studying the microscopic anatomy of chicken oocytes, Purkyne identified a distinct rounded structure within the cell, what he called the germinal vesicle. This structure was in fact the nucleus, and his discovery confirmed that animal cells, like plant cells, also contained this central feature, and based on the nature of the cells he was examining, Purkyne suggested that the nucleus might play a role in development and organisation, hinting at its fundamental biological importance. Now later on in his career, Purkyne went on to coin the term protoplasm, referring to the substance within the cell that surrounds the nucleus, what we now recognise as the cytoplasm containing the various eukaryotic membrane-bound organelles. He proposed that this material was essential for maintaining cellular activity. And so moving into the early 19th century, we meet the Scottish botanist Robert Brown, a figure whose careful observations would further cement the importance of the nucleus in cell biology. Now, Brown is often remembered for describing Brownian motion, the random movement of pollen grains suspended in water, but his most significant contribution to cell theory came a few years later in 1831. And so whilst he was examining orchid tissue under the microscope, Brown noticed a distinct dark, rounded body within each cell. What stood out to him was its consistency. This structure appeared across a wide range of plant species and was clearly an integral part of the cell, not just a visual artifact. And so Brown formally named this structure the nucleus, with his work representing the first clear and systematic description of the nucleus as a universal cellular component. Now by this point in time, several scientists had seen the nucleus. Baur had illustrated it in plant cells, Purkyne had recognised it in animal cells, and Brown had formally named it. But despite these discoveries, its structure and function remained a mystery. And it wasn't until around 1838 that a potential function was finally proposed. Some scientists of the time suggested that the nucleus might be responsible for the formation of new cells, referring to it as the cytoblast, literally meaning cell builder. Now, although this interpretation wasn't entirely accurate, it marked an important step in shifting scientific thought from simply seeing cellular structures to beginning to ask what they actually do, a question that would drive much of cell biology in the decades to come. And so moving forward into the 1870s and 1880s, we hit a really exciting period where there were a series of rapid discoveries. Scientists all over the world were now studying cells, often independently, using both plant and animal material, and yet they were all uncovering similar patterns. And what started to become clear was that the nucleus wasn't just sitting there, it was doing something vital. Researchers began noticing that during cell division, the contents of the nucleus seemed to be shared between the two new daughter cells. That led to a big realization: the nucleus must be involved in heredity. It was somehow responsible for passing information from one cell generation to the next. And right around this time came another major breakthrough, the discovery of mitosis. Being able to actually see the nucleus divide under the microscope helped scientists understand its role more clearly. It showed that the nucleus was actively involved in organising and distributing genetic material. In other words, it was the genetic carrier of the cell, which was a huge step forward, marking the point where cell biology and heredity started to truly come together. Then, as we move into the early 20th century, things really started to come together. Scientists were beginning to put the puzzle pieces together, connecting what was happening inside the cell and how traits were inherited. Around this time, Mendel's work on genetics, which had largely been forgotten since the 1860s, was rediscovered. His principles of inheritance suddenly made perfect sense when viewed alongside everything researchers had learned about the nucleus and chromosomes. This led to what became known as chromosome theory of inheritance, the idea that genes are carried on chromosomes and those chromosomes are housed within the nucleus. And so for the first time, scientists had solid evidence linking the physical structures inside the cell to the transmission of hereditary traits. So, it was now proven that the nucleus is the carrier of genetic information, not just a structural feature, but the command centre for heredity itself. This discovery completely reshaped biology and paved the way for the molecular research that would follow, ultimately leading to the discovery of DNA and the genetic code. And with that, we come to the end of this video. I hope you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.